Hi, this is the second part of phylogenetics, and it's covered in chapters 24 through 25 in integrated molecular evolution. So I'll talk a bit about the methods used in phylogenetics as well as some variations at the end. If you start with a hypothetical tree, this one has a certain structure so that the distance between the node and A, that branch length is 1. The branch length to B is 2, and then there's an internal branch of length 2 and a branch out to C of length 4. So we're going to see how well these different methods do in coming out with this tree given the data that can be derived from this. So working backwards, we make a data matrix based on this tree, and then we'll use that data matrix with different methods to see if we can reconstruct this same tree. So here's the data matrix. The distance from A to B is 4. The distance from A to C is 7. The distance from B to C is 9, and it's just repeated on the diagonal. So this first numerical method is called UPGMA, which is unweighted pair group analysis using arithmetic averages. So what is done is you take each one of those distances from A to B, B to C, etc., and you come out with an arithmetic mean for the two branches from that. So in this case, the two closest taxa are A and B, that they have a distance of 4, so 4 divided by 2 means that each branch will be of equal length of 2. It then goes back to the original matrix and recalculates based on the distances that have been determined. From the node to A is 2, the branch length is 2, and the branch length to B is 2 also. So the new branches then are 4, and then it calculates the distance from A to C and B to C. They're not always integer numbers at this point also. So in doing this, then it comes out with a tree that looks like this, the final UPGMA tree. And if we compare it to the original true hypothetical tree, we see that it differs in certain ways from the original tree, so that the branch to C is correct, and the internode, or the internal branch of length 2 is correct. But the final distances out to A and B are incorrect. The totals are correct. It's 4 steps from A to B, but the branch lengths are not the same as the original tree. So it does an okay job, but not a perfect job. So here's a second method called neighbor joining. It not only measures the distance from one taxon to the next, or one OTU to the next, but it remembers in a certain way the distances to the nodes. So it begins with a star diagram. We only have 3 OTUs in this case, but you could have as many as, thousands making up the star. And it first joins the 2 closest taxa together. And this one remembers the distances to the nodes by doing all the calculations ahead of time. The distance from A to B, A to C, B to C, etc. So that it knows that it's a little further from B to C than it is from A to C. So it moves the node over in this case. So when it joins the first 2 taxa together, which are the closest, A and B, it knows that the distance between A and the node is 1, and the distance between B and the node is 3. And then it joins the third taxon up. So in this case, then it joins C to a node and that internal branch of 2, and comes out with a tree that looks a lot like the original tree. In fact, the distances are identical. So both neighbor joining and UPGMA are very fast methods. That is, they're computationally fast. But neighbor joining is much more accurate than is UPGMA. So for large data sets consisting of hundreds to thousands of taxa, neighbor joining has become the first choice for most studies now. The next method I want to discuss is maximum parsimony. And this is what's called a discrete method. The other two methods, UPGMA and neighbor joining that I just spoke about, are numerical methods. That is, they do a calculation. Maximum parsimony looks at the number of steps. So it begins first by joining the taxa in every possible way that it can. So if you join 4 taxa together as rooted trees, there are 15 possibilities, and these are the 15 possibilities down here, the different branching patterns. So maximum parsimony then examines each tree according to the characters and then counts up the number of changes that have to occur in that tree to get to the final result. And so up above is the character matrix. It's just ones and zeros, meaning presence or absence of the particular character. So D has none of the characters, so you can put that down at the root. In the first one up there, to get to C, you have to add character 4 and character 2. So that's the line out to C. To get to B, you have to add characters 1 and 3. So then you have characters 1, 3, and 4. And finally, adding character 2 to get to A. In the second phylogram, the second one, the second tree to evaluate, similar changes, but they're slightly different to get out to B and then get up to C and finally up to A. So that in the tree A, there are 5 changes and 1 homoplazy. That is, character 2 has to be added on 2 different branches. The other one on the right, the tree on the right, entails 6 changes, and there are 2 homoplasies. So characters 1 and 3 have to be added to branches 2 different times. So the tree on the left has both fewer changes and fewer homoplasies, so then it's chosen as the most parsimonious tree in this case. Because it joins the taxa together in every possible orientation and then tests each one, then it builds up very fast to a large number of trees that have to be evaluated. So as far as computer time, as you work your way up into 10, 20, 50 taxa, the computer slows down. There's a lot more computation involved. So you can see the number of trees to evaluate increases greatly here, so that if you have 3 taxa to join, there's only 1 tree to evaluate. 4 taxa, there are 3 trees to evaluate. Those are the 3 at the top. For 5 taxa, there are 15 trees to evaluate, but by the time you get to 10 taxa, there are over 2 million trees to evaluate, and by the time you get to 50 taxa, then it's 3 times 10 to the 74th trees to evaluate, which is much more, many more than just a little desktop computer can handle. So for small data sets, maximum parsimony works very well. It's highly accurate. But for larger data sets, it's just too computationally costly to use this one. So for large genomics comparisons, then neighbor joining is normally used. There are several search strategies for maximum parsimony and other programs also, including maximum likelihood, which I'll discuss next. The first one is an exhaustive search. So that in the case of 50 trees, examining 10 to the 74th trees is what it would have to do, which is way, way too many to do. The third one down there is a heuristic search. And so, as I said, it first builds all the possible connections, the trees in all the possible connections, and then starts evaluating those. In the case of a heuristic search, it just selects a random tree, one of the random ones that it's already put together, and then starts from there and looks for a local minimum, that is a more parsimonious tree that surrounds the one that's there, and keeps moving out until it finds a most parsimonious tree in that area. So in many cases it'll find the correct tree, but in some cases it will not be able to find those. The second type of search is branch and bound, which is called a semi-exhaustive search technique or strategy. It starts with a random tree or a chosen tree, you can choose one, and begins moving up a local branch of a search tree. So I'll be talking about a tree, the phylogenetic tree, but there's also a search tree. So it's going through searching each one of those trees for a local minimum. And it also searches branches further out from those branches. If they are already longer, then it eliminates entire regions of those trees to search through. And I'll show you that in the next series of slides. So here's what I mean by the search tree. So the search tree is going through joining one taxon at a time, building out the number of phylogenetic trees that it will have to search. So there's the ABC trees, and then there's the ABCD trees, and then there's the ABCDE trees, all the possible ones, organized in a way that joins one taxon at a time. The first thing it does is it moves out on that search tree to one of the final branches at the end, and it then calculates the number of steps based on the data that it takes to build that tree, that particular phylogenetic tree. It retains that tree and then goes one step back on the search tree, and then sequentially examines each of the phylogenetic trees out from there, and calculates whether they are longer or shorter than the tree that it's held in memory. If one of those is shorter, then it'll retain that in memory. But if they are all longer, then it eliminates those as being less parsimonious. Next, it proceeds back one more step and then goes down the next branch of the search tree to examine the next tree in line, and determines whether it's longer than the tree that it's holding in memory as the most parsimonious tree so far. Now, if that one is already longer, and so it eliminates that, logically the ones beyond that are also longer, probably much longer. So rather than even examining those ones, it doesn't even examine those, it just eliminates those from further searching. And so it saves a lot of time there by eliminating a lot of trees that are obviously longer, and it would not have to search through those. Next, it sequentially again goes down, goes back up the tree and then back down by one, testing the next tree to see whether it has fewer steps in it than the most parsimonious tree so far. And again, if that particular tree is longer than the one that's held as the most parsimonious tree so far, that's eliminated, and everything beyond that is also eliminated without even searching through those. So the final result is that it goes back to the one that it found was the most parsimonious of all the trees in the phylogenetic tree search set that it was looking through. And again, this one's called semi-exhaustive because it does come out with the same result as an exhaustive search would do, but it's much, much faster. Often statistical methods are used to examine each one of the branches and determine the support for each branch based on the data. One of those methods is bootstrapping, and it's a resampling method with replacement, and I'll explain that in a few minutes. If you have a set of characters and the OTUs or taxa, you can build a tree based on that data. But to test whether that tree or any other tree is well supported by that data, resampling is done. So a new matrix is begun by resampling some of the data. So it'll randomly select one of the characters to choose to build a new matrix. In this case, character 9 is the initial one that is being used. Now, by replacement, it means that the data set still has character 9 and it can be chosen more than once. So here a second character has been chosen at random, character 2, so that's put into the matrix. And then a third character is put into the matrix. You'll notice it's character 9 again. That's what I mean by resampling with replacement. Character 9 was replaced in the original matrix and was chosen again at random. And then more characters are chosen. Again, you can see 7 and 2 were chosen twice, again because of resampling with replacement. And now a tree is built from that matrix. And so now you have 2 trees, the original tree, tree 1, and now a second tree built from a resampling of the data. And then a third tree is made, starting a new matrix, again selecting characters at random. And usually in most bootstrapping, it's done 500 to 1,000 times. So 500 to 1,000 trees are then produced, and then the data, or that information, is searched to determine how many times particular branches appear on each one of those trees. So if a branch occurs in all the trees, then it's supported by 100%. If it's only there in 50% of the trees, then it's only supported by 50%, and usually 50% is the cutoff value for counting it either as supported or not supported. Here is a tree that we built based on some sampling from some glacial ice samples. So you can see there are numbers on each one of these branches. There's a branch here supported by 100%, 100%, 97%, 78%, etc. Branches that have no numbers on them mean that the support was less than 50%, meaning that they aren't very well supported at all by the data that was used. In this case, ribosomal RNA sequences. Another phylogenetic method that's pretty powerful is maximum likelihood, and it's used quite a bit also, although it is computationally heavy because what it's using is probabilities and tests all of the possible trees again, or all the possible branches and the trees. So essentially, it's looking at the data, looking at the number of A's, G's, C's, and T's in the data that you send to it, which is sequence alignments, and then it asks what are the probabilities of this sequence A, G, C, T, C, C, C changing to A, G, C, T, 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 T. And so it'll look through and determine it has to have 3 transitions, C to T changes, and what are the probabilities of that. It does that for each branch that it joins together, and then determines the log likelihood, the natural logarithm of the likelihood for that particular branch. And for the entire tree, then it's the sum of the log likelihoods of each one of the branches added together. One of the uses of maximum likelihood is that you can test hypotheses with maximum likelihood. So if you get a certain result in a tree, you can also compare it with another tree. Let's say you want to put, you're looking at human evolution and you say that chimps are closer to humans than are gorillas. But then you want to have a second tree to say, well, what if we put gorillas closer to humans than chimps? Is that less likely or more likely? So you can test these different hypotheses. So should number one, should taxon A be on the same branch as taxon B? The maximum parsimony tree is congruent with the maximum likelihood, or gene A is in the same family as gene B. Several years ago, we did a study where we used maximum likelihood to test some hypotheses. So this was looking at the genus Dermaceae, which is a mushroom, and comparing it to the genus Cortinarius. So Dermaceae is on the top here, and Cortinarius is on the bottom. And then looking at the sub-classifications of this, the section sanguinii, section malacori, etc., to determine whether those were supported by the molecular data. And so we tested in several different ways and actually came out with a result that was surprising in some ways, in that the genus Dermaceae was well defined, although the subgroupings were not as clear, and some of them were not supported by the molecular data and the maximum likelihood analyses. Also, so we proposed the classifications on the right, which ultimately were accepted, actually. So also there was one down here, Dermaceae olivacea picta, which was down within the Cortinarius clade. And so it obviously, we think anyway, is misclassified and should be a Cortinarius rather than in the genus Dermaceae. So the way these tests were done, the evaluation of these different trees is the maximum likelihood tree here consisted of a total tree length of 1,051 steps, and it's just called the best. And then there's the lawn likelihood. And then that was compared with the maximum parsimony tree, which had 3 fewer steps and had about the same lawn likelihood with a low standard deviation. So that was not rejected as a hypothesis. We did another constrained tree that was also congruent with the maximum parsimony tree, just to put all of the certain types of Dermaceae together. That was also well supported and was not rejected by maximum likelihood statistical tests. But then we moved things around, constrained different trees. So that is, we would make sure that certain taxa were joined together in the tree. So they were somewhat constrained in the way that the rearrangements could be made. And 3 different constrained trees were worse than the maximum likelihood and maximum parsimony trees. So those were rejected. And so those hypotheses, moving certain taxa around, joining certain taxa together, were rejected. And that led to that final well-supported tree that I just showed. The last method I want to talk about is Bayesian phylogenetics. And it has some similarities to maximum likelihood because it deals with probabilities of joining certain sequences and looking at certain changes in between taxa. So it begins again by joining the closest sequences together and then works outward from there, which is how maximum likelihood starts also. Then it starts with a model and tests whether the data are consistent with the model, comparing observed distances with the calculated distances. And then it uses what's called Markov chain Monte Carlo methods, looking at whether the change in the first state, one tree, is more likely to occur than the change to the other tree, and tries to find local minimums and move towards more and more likely trees that where the changes in those trees is more likely to occur than the current state that it's comparing to. So it's comparing the current tree with a tree that has some changes in it and determining whether those changes lead to a better outcome from probabilities than the current tree. So it's as statistically robust as maximum likelihood, but one major advantage is that it takes less computer time, although it still is fairly computationally heavy, so it takes a lot of computer time. So in choosing a method, UPGMA and neighbor joining are fast and easy, but neighbor joining is by far superior to UPGMA. Moving down, maximum likelihood and maximum parsimony and Bayesian are all good to excellent, give very good results. Although maximum likelihood is the slowest one, and it's a little difficult to use. The other ones take less time, and they're also, moderately easy to difficult to use. As far as the analysis of genomes now, neighbor joining is used initially because it's very fast, but if people want very accurate results, they are now using Bayesian methods to a greater extent than the other ones. They are available in several different software packages. So NITSIS, which is Numerical Taxonomy System, is for the PC only, costs about $130, and it has UPGMA, neighbor joining, and a few other statistics. Primarily the data types are presence or absence, so it's not as useful for molecular data such as sequences, DNA sequences, RNA sequences, or amino acid sequences. Mega is, I think it's up to Mega 8 maybe version. It's very good and it's free, and it's for Windows, DOS, or Macintosh. It has UPGMA, neighbor joining, and maximum parsimony. I don't know if the newest method has Bayesian. And it's very useful for nucleic acids, amino acids, as well as binary 1-0 type data. Phylip is a phylogeny inference package, primarily for DOS, but it's also been written for Windows, Unix, and Macintosh. It was mainly written for maximum likelihood, but it also includes maximum parsimony and others now. Primarily used for nucleic acids and amino acids, but can be used for binary data also, and it's free. There's the source for that. PALP, phylogenetic analysis using parsimony, was mainly for parsimony at the beginning. The price varies depending on which platform you're using, but it's mainly, again, for maximum parsimony, but has maximum likelihood, neighbor joining, UPGMA on it now. Very powerful for nucleic acids and amino acid types of data sets. And finally, Bayesian. It's mainly, there may be other packages, but Mr. Bayes was the original one, works well with Macintosh, Windows, and Unix. And it's Bayesian calculations and hypothesis testing. And again, it can be used for nucleic acids, amino acids, restriction site data, and morphological data as well. And it's free online, so you can download that and start using it. So as far as those packages, here are the different programs, different algorithms in each one. NITSIS, Mega, Phylip, PALP, and Mr. Bayes. The utility of each one of those, especially Phylip, PALP, and Mr. Bayes, are high. Mega, I think it's moderate to fairly high. It's a good system. Ease of use, is very easy, palp's very easy. Phylip I find a little more difficult, although some people find it easy. And Mr. Bayes is a little bit difficult to learn at the beginning. Once you learn it, it's not so bad. So I talked about the main types of phylogenetic analysis. Now I want to branch off into a couple of other areas. One is into phylogenetic networks. So in a standard phylogram, you have nodes and branches, just straight lines joining the taxa together. In phylogenetic networks, there are splits and edges, and things are joined together in networks rather than in straight lines. Some researchers began developing these phylogenetic networks because the regular phylograms at times gave unresolved trees, and so it was difficult to make a tree from a certain data set or a resolved tree from a certain data set. And there were actually some reasons for this. So there's recombination, there are sometimes horizontal gene exchanges and things like that. And so you can get from point A to point C or point B in multiple ways, depending on what sorts of molecular mechanisms have occurred in between those two taxa. So it is indicating that the joining of particular branches together is sometimes more complex than just a simple line. So here's one even a little more complex, where there's some lack of resolution in the FDE part of the phylogram. But if you use a network algorithm to solve that the particular joining of those taxa, then you come out with a network where there are several possible routes that have occurred or may have occurred to lead to each one of those taxa on the terminal branches. I'll just show one example. This is a phylogenetic network for blue tongue viruses, which infect domestic and wild animals, and was done primarily because of an outbreak in sheep, I think in Bulgaria or something like that. But you can see that rather than having straight lines, there are multiple splits and edges joining these taxa together, probably indicating some recombination events as well as some horizontal gene transfers among these viruses at different times in the past. So one thing that can lead to these networks and in standard phylograms can lead to some confusion are horizontal gene transfers. So here at time 0 you have two species, and then at another time at T1 there's a transfer of some genes from species A to species B, and then look at them, examine their sequences at a later time, and find that for most genes, there's a separation between A and B prime, that other species. But in other genes, there's very little change among the genes. And so in some cases, that would confuse phylogenetic analyses. So here's an example where you have analyzed two different genes. In gene A, you get one branching pattern, and then in B, there's a very different branching pattern because there's been a horizontal gene transfer of a particular gene in the species F there, so that it looks like it's misplaced with respect to the other taxa. Now, for standard phylogenetic analysis, then this could be a problem, but when you're analyzing many genes and use phylogenetic networks, then you can reconcile this through the network changes that can be seen as splits and edges. Other things that can affect phylogenetic analyses and phylogenetic networks can sometimes pick these up are duplication, deletion, mutation, and recombination events. And so you can have genes being duplicated, you can have genes being deleted. There are intragenic crossover events that can lead to a chimeric gene. The duplications can lead to different orthologs as well as paralogs. And then you can get gene exchange among between chromosomes and between taxa, and then translocations as well of translocations and transpositions of genes. So that instead of getting straight lines, if there's been a lot of recombination, a lot of duplication, things and other things that happen in the genome, then the phylogenetic networks often can reconcile those to a greater degree than just doing standard phylogenetic analyses. With phylogenetic analyses, you can follow characters around, follow duplications, mutations, etc., and infer those and actually map them onto the trees. So in this case, along one branch, there are some duplications, followed by mutations, followed by more duplications, some losses in the genes, and formation of pseudogenes, and those can be mapped on those trees, and then you can actually, place them in time as to when they have occurred or might have occurred. And on the other branch also you see mutation, duplication, more mutation, losses and formations of pseudogenes on that line as well. And so you can plot those on these trees also and come out with a sequential change pattern in those different taxa. Phylogenetic methods can also be used to study hybridization and endosymbiotic events. So in this case, the species A has diverged into species A and B. At a later time, some of those individuals join together to form a hybrid C. So now you have A and B as well as hybrid species C in the middle. After the discoveries that the mitochondria were originally proteobacteria that entered as endosymbionts into the first eukaryotic cells, and that cyanobacteria later entered certain cells to become the chloroplasts, we've started drawing trees a little differently, so that on the left here you see that the origination of eukaryotes were because of these movements of these cells, one into another, and so all those genomes are mixed together as well. But a different way of drawing that is over on B, where now the eukaryotes are really the product of joining of an archaeal cell with a bacterial cell, and probably more than one species is involved, one to help form the original eukaryotes and then another one to become the mitochondrion. And then later, cyanobacterium entering one of those cells to become the first plant cell with a chloroplast. Now, as I indicated in previous lectures, there were more endosymbiotic events to form more organelles as well. And then there have been some theories as to the origination of diderm bacteria. Those are gram-negative bacteria with two membranes, versus the monoderms, the gram-positive bacteria with one membrane. So if you look at a phylogenetic tree, then you find that there's a mixture of monoderms and diderms sort of all mixed together. So these monoderms and diderms then are said to be polyphyletic, meaning that they exist in different parts of these phylograms. But a few years ago, a researcher looked at certain genes in the monoderms and then tried to to determine how those monoderms came together to create diderms. And in doing so, he showed that if a Clostridium and an Actinobacteria were joined together, they could form the diderms, and then the diderms would be monophyletic. There were some problems with this analysis, and so people haven't fully accepted it yet. One problem that I've always had with phylograms is that most of the diderms, the Proteobacteria, Chlorobi, etc., pretty much group together as a monophyletic group, not completely. And the monoderms group together in a monophyletic group. But then Cyanobacterium is way off on its own. Also, cyanobacteria are unique in that they have both photosystems, reaction center 1 and reaction center 2, which eventually become photosystems 1 and 2. Whereas all of the other diderms have only one photosystem, either RC1 or RC2. So how can you build a model, a phylogenetic tree, that keeps those things in agreement with one another? And so I put together a little bit of an analysis here on this slide, and I'll try to explain this. In most phylogenetic trees, monoderms come out as a monophyletic group, and they do here as well, as a Firmicutes, Chloroflexi, and Actinobacteria on the lower right side of the diagram. Those are all the monoderms. The diderms usually come out as polyphyletic. That means they are in different parts of the cladogram and not together. So I looked at photosynthetic apparatus, starting down with the brown little square. Down there, the ancestral reaction center was duplicated, and then you have it diverging into the blue, which is the reaction center 1, and the red, which is reaction center 2. To get a cyanobacteria, they have both of those reaction centers, so those had to come together to yield that purple group, cyanobacteria, with both reaction center 1 and reaction center 2. And one of the reaction centers, reaction center 2, also gained a manganese, which is the oxygen-producing part. So that's when they became oxygenic. All others are non-oxygenic photosynthesis. So I also plotted numbers on there, and each of those numbers, 1 through 12, indicate either insertions or deletions in certain genes that are characteristic of those groups. And so those go in order according to what other people have found also evolutionarily. So that's consistent. I also mapped nitrogen fixation, and nitrogen fixation is lost in many of the groups and retained in others, as shown by either NF plus or NF minus, and how many nitrogen fixation genes there are, or genes involved in nitrogen fixation. And from the Firmicutes all the way up to the Proteobacteria, they almost always increase, or nitrogen fixation itself is lost in those groups. A couple of the groups that don't fit into this are the Deinococcus thermus group, the DTF, and the Negativicutes, and one other group that are normally grouped with Firmicutes, but they have been recently split off from those. The only way to explain those is if you invoke 2 more endosymbiotic events to get the 2 membranes, and so that's what those parts of the phylogram stand for. So phylogenetics can be used to attempt to at least answer some questions by mapping certain characters onto some basic trees. During the past few decades, A new part of phylogenetics has arisen, and that's phylogenomics. And so this is where you compare genomic regions from several different organisms. And normally what you find is the mapping of those different sequences is in different locales on each of the genomes. So then you have to concatenate those sequences in the computer in order to align them properly. One of the reasons that the chromosomes themselves don't line up in most cases, although in some cases when the chromosomes do line up, that is, they're synchronous or they're in synteny, is when they're very closely related organisms. That's when you can get fairly good alignment. But over evolutionary time, then, even though you have the same genes, many of them or most of them are reorganized on the chromosomes, and it doesn't seem to affect their expression as much as you might think, that they're still expressed. They're just in different locations on the chromosomes for those organisms. So here are just some of the things that can happen. If you start with a chromosome set as in A, you can have splitting of chromosome pieces. You can have parts of chromosomes recombining to form new versions of the chromosomes. You can have splitting of the chromosomes, joining of the chromosomes, almost anything that you can imagine can happen. You can even have a piece like the green region on the right-hand side that doesn't change position much. And so in many cases, the sex chromosomes stay pretty much intact through many different changes during the species, genera, and families. Here's just a general example. If you have genes in genome A and genes in genome B, a certain number of genes will be in common between those. And you can pull those out one at a time and align them and concatenate those sequences from genome A and genome B so that you have the analogous genes being aligned, and then do phylogenetic analyses on those. As more genomes are added together, then the number of common genes usually is reduced because you're trying to line up a larger number of disparate genomes. And so the common genes still can be concatenated, all concatenated in the same way, aligned, and then phylogenetic analysis or analyses can be done on those aligned sequences. So here's a comparison that was done several years ago, just comparing a eukaryotic genome with an archaeal genome and a bacterial genome. I think the eukaryote was Saccharomyces, the archaea was Methanococcus, and the bacterium was E. coli. And you can see that 65% of the genes are in common with all of those. So even disparate species, Saccharomyces and E. coli, can be aligned. You can get analogous genes and line those up and do phylogenetic analyses on them. Here's another comparison that was done a few years ago, comparing 3 animal sequences, that is Cenorhabditis elegans, which is a nematode, Homo sapiens, and Drosophila melanogaster, compared to a yeast, which Saccharomyces cerevisiae. And so in that data set, 25% of the genes were in common to all 4 genomes, and 42.9% were in common among all of the animals. And so you can do phylogenetic analyses on a fairly large proportion of each one of those genomes by joining them together in a sequence alignment. Here's another way to compare genomes, and this is a study done on Buchnera aphyticola, which is a symbiont of sap-feeding insects. Sap is nutritionally very poor, but these bacteria grow inside of the sap-feeding insects and produce amino acids and cofactors, vitamins, and things like that, so that sap-feeding insects can actually survive by feeding on the sap. So as you go from the bottom to the top, different genes have been found to be in common. The ancestral genome consisted of more than 638 genes. As you move up, some 499 genes are in common to all strains. And you can see the strains above. Some of them are in sharpshooters, some of them are in aphids, and some of them are in other sap-feeding insects. As you move up on each one of these lines towards that species, you can see that 64 genes have been lost leading up to one of the varieties, where those 64 are actually still functional genes in the other line. And then you go up a little further, 8 have become pseudogenes in that one, and the same 8 are still functional in the other line. And then 2 have been lost along the left branch, and the same 2 have become pseudogenes in the right branch. And you can work your way up to the next split, where 22 are still functional in that middle branch, where those same 22 have become pseudogenes, working all the way up to each of those 3 varieties of this Buchnera. So you can use those genomic sequences to trace these changes within these varieties as they evolve along with their hosts. Here's a comparison that goes along with the phylogram I showed earlier. So here's a comparison between Shigella, E. coli 0157, so both of those are pathogenic, and the E. coli K12. And you can see 59%, almost 60% of the genomes are similar. That is, they have analogous sets of genes, whereas E. coli has 7.7% are unique to E. coli K12, 12.9 are unique to 0157, and some of those are the genes that cause severe diarrhea and pathogenicity in humans, and then 3.6 are unique in Shigella. And finally, this is a study that was done in higher plants a few years ago. So Oryza sativa is rice, Zea maize is corn, sorghum bicolor. Those 3 are all grasses, so they're relatives of a sort. They're in different families and things, but they're all grasses. And Arabidopsis is not a monocot, it's a dicot, so it's very different. But in the middle, you can see that 55% of the genes, gene families and gene versions, are similar among all those plants. And those 2 groups diverged about 40 to 50 million years ago. Then you add another 13.5% that are in common with all of the grasses as well. So that makes a total of about 69% of the genes among the grasses are in common to all of those grasses. And in fact, among those, 7.2% of the genes are unique in rice, 3% are unique in Zea maize, 1.7% are unique in sorghum, and 6.9% are unique to Arabidopsis thaliana in this study. So that's the end of the phylogenetics and phylogenomics section. This will then get us all up to speed so that now we can start talking about genomes, starting with virus genomes and working our way up. So next time will be Genomics Part 1, and it will be a several-parted set of presentations. Talk to you next time.